Now starting up with the next segment, I request all the dignitaries to kindly proceed towards the lamp lighting ceremony.
Today is the time to get updated about some aspects of the molecular biology. So I think we will get benefited by this symposium. With this, I want to call my principal sir, who is the first person behind this symposium, so that he share some words with us before starting the scientific session.
Respected Dr. Punita, the speakers of the function, Dr. Vicky Mehta sir, dear scientist PRI, Dr. Vijay Kumar, Dr. Sudhakar, Dr. Yun Singh, Dr. Anil Kumar, Dr. Musa, the colleagues and the participants of the seminar. Today we are going to discuss many problems and advancements in the molecular biology. The efforts of Dr. Chaudhary and Dr. Anil Kumar in making this and starting the procedures in the Central Research Lab. I hope that all the deliberations of the seminar will be useful to all the participants and we all will be enlightened with this symposium. Thank you.
I request Dr. R.S. Actuary of Microbiology Department and Research Center Lab to please come and give a brief introduction. Before I request ma'am to please come, I would like to tell something about her. She has completed her MBBS degree in an esteemed institution in Karnataka with a gold medal in surgery and has Scientific. She has been invited as guest speakers and organized and conducted courses for NAPN and NAPH in our institute. She is the Associate Guest Editor of Drama Journal of Medical Sciences and Review and Advising Member of various journals and a coordinator of NAM. Ma'am, please come and say a few words.
It gives me immense pleasure to welcome one and all to this great symposium. Respected principal sir and all the dignitaries present on the dais and off the dais, I have been given a task to introduce molecular biology to all the delegates. Life is a transition from biology to molecular biology. Are you ready for this transition? So after this brief introduction, I think you will be very curious to know the various aspects of the three domains of life. Current research theory supports the division of living organisms into three domains: the bacteria, the eukaryote, and the archaea. So, the many cells are being categorized based on the complexity of their cell divisions. So, most of the cells are organized into either the prokaryotes and the eukaryotes. But whatever is the cell, the basic DNA and the chromosome is the DNA or the RNA. What is molecular biology? The attempt to understand biological phenomena in molecular terms, or the study of gene structure and function at the molecular level to understand the basic heredity, its variations and expression in patterns of genes. As a result, it is a study of molecular basis of the process of replication, transcription and translation of genetic material. Molecular biology mainly concerns itself with the understanding of interactions between the various systems of a cell, including the interactions between DNA, RNA and protein biosynthesis, learning how these interactions are regulated. This field overlaps with other areas of biology and chemistry, particularly genetics and biochemistry. It is a joining of aspects between genetics and biochemistry. Components involved in molecular biology. All life depends on three critical molecules, that is the DNA, the RNA and the protein. Inside a living cell, we have four life alphabets: A, T, G and C, like similar to the English alphabets, which are 26 letters. Now, if we see from these 26 letters, we can make volumes of books and chapters. Like this, life alphabets are organized into chromosomes and the DNA and the gene. So, though these structures are all static, but their functions are dynamic, which carry out various functions of inheritance. A brief history about molecular biology. Many scientists earlier in 1950 and early 60 have characterized the molecular components of cells and organisms into just the storage of genetic information, the RNA and the proteins, the major structural and enzymatic form of molecules in cell. Coming to the journey of this molecular biology, to start with, if you see the microscopic biology, which began when Sir Robert Hooke discovered the cell. Then Matthias Sheldon and Theodor Schwann, they studied the details of the cell. Gregor Mendel, who is called the father of genetics, who discovered heredity and described as the two alternate hereditary units of dominant and recessive trait. Sir John Friedrich, who discovered DNA and named it nuclein. So earlier DNA was called as nuclein. Then Edward Zaya showed chromosomes are composed of nuclein. In 1899, Richard Altman renamed nuclein to nucleic acid. By 1900, the chemical structure of all 20 amino acids had been identified. In 1902, Emil Hermann Fischer won the Nobel Prize because he linked amino acids to form the protein. And Thomas Hunt Morgan discovered genes on chromosomes are the discrete units of heredity. 1911, Fiobo's R. Theodor discovered RNA. And 1941, George Baden discovered that genes make proteins. And 1950, an important discovery by Edwin Chabot to find that cytosine, guanine, adenine and thymine complement each other. It's a major history. And in 1950, Melbourne Bush isolated tRNA and Alfred Herschel isolated genes from DNA. And 1952 to 53, two important scientists, Watson and Crick, deduced the double helix structure of DNA. In 1956, George Emil Palade showed the site of enzyme manufacturing and discovered the ribosomes. And 1970, David Baltimore independently isolated the first restriction enzyme called the endonuclease, which is the key point for the genetic engineering today. So the DNA can be cut into reproducible pieces and it can be linked into the different vectors like bacteria and produced in enormous number. This is genetic engineering. 1977, Philip Sharp and Richard Roberts demonstrated mRNA. And 86, the sequencing mechanism was introduced. In 1986, Human Genome Initiative was announced. And in 1995, resolution maps of chromosomes were started. In 1995, George Drake Venter first sequenced the bacterial genome, which is a recent and latest topic. Sequencing is a very important topic. And 1995, automated fluorescence sequencing instruments and robotic operations were introduced. In 1996, first eukaryotic genome is sequenced. And 1999, first human chromosome number 22 was sequenced. And 2001, International Human Genome Sequencing and first draft of the sequence of human genome was published. In April 2003, Human Genome Project was completed and mouse genome was sequenced, rat genome was sequenced in 2004 and next generation sequencing genome being sequenced by a dozen later on. So after this brief introduction of history, some terminologies commonly used in molecular biology is nucleic acid, the gene, the genotype and the phenotype. Why we must be familiar with molecular methods? In many developed and developing countries, diagnostic methods are switched on to molecular methods and no scientific journal is willing to accept or publish any article without incorporation of molecular methods, and it's a mandatory for all the PhD students now. Antibiotic resistance is a growing concern now, and until and unless we know the molecular identification of any organism, it is very difficult for us to contain the spread. How does a molecular diagnostic work? Every organism contains some unique species-specific DNA sequence, except in some virus, which is RNA. Molecular diagnostic makes the species-specific DNA visible. And this is the DNA structure, which has got the purines and the pyrimidines linked by the disulfide hydrogen bond. So adenine binds with thymine and guanine with cytosine. And except RNA is very similar to DNA, except that instead of double-stranded, it is a single-stranded, and instead of thymine, it is uracil, and the sugar is deoxyribose instead of deoxyribose. Now, the central dogma of molecular biology lies in this slide, where the DNA is the transcription and the translation. So, DNA is converted into mRNA, and you will get the protein synthesis. Now, when we really need molecular methods, molecular diagnosis is most appropriate for infectious disease that are difficult to detect, identify, or test for susceptibility when the conventional methods don't come in hand. The applications of molecular biology, it's used in research, it's used for diagnosis of infectious disease, it's used in transplantation, identification of meta-forensic analysis, as used as gene therapy for incurable diseases and drug design. Molecular methods are necessary if the traditional methods provide poor results, if the microscopy fails, if it is an intracellular organism, if it is having a low sensitivity, the seropositivity is common, and subtyping is mandatory, and the microbial growth, if it is very slow, especially in case of Mycobacterium tuberculosis. The molecular diagnosis, some of the common molecular diagnostic methods are grouped into either hybridization, amplification, sequencing, enzymatic digestion of nucleic acids. Hybridization is a process whereby two single strands of nucleic acid come together to form a stable double-stranded molecule that will bind and stay together only if the sequence are complementary to each other. So this is the diagram which shows the hybridization technique of the probe as well as the target. So the detection of hybridization can be by radioactive method or biotin adenine receptor or chemiluminescence. Detection of hybridization can be in a solution format or solid format by filter hybridization, southern hybridization, sandwich hybridization and in situ hybridization, especially when you have to detect a pathogen in the tissue. This is the hybridization in the solute format and solid support format. We have three methods: filter hybridization, southern hybridization and sandwich hybridization. Next, coming to the amplification method, we can amplify the nucleic acid or the nucleic acid probe can be amplified or amplification of the probe signal. Now, amplification of the nucleic acid is also called the polymerase chain reaction, which has the three main steps: the denaturation, the annealing and the extension. This small animation will tell you the three different techniques: the denaturation, which occurs at 94 degrees, annealing at 54 degrees and extension by the polymerase enzyme at 72 degrees centigrade. These three steps occur every 3 to 5 minutes and it is after around 2 to 3 hours. We can see that there is around after 50 cycles, one molecule of DNA will be exponentially raised to 10 to the power of 6. Detection of PCR products can be done by gel documentation, ethylium bromide, southern dot analysis, polygonal restriction, polygonal hybridization, reverse dot technique. This is dot analysis and the gel documentation, which is usually done after the polymerase chain reaction. And then we have derivatives of PCR methods: the multiplex PCR, the nested PCR, the quantitative PCR, the reverse transcription PCR, the arbitrary PCR and the real-time PCR. This is a multiplex PCR where different strains and different primers are together added in one Eppendorf tube and the results are identified. In the nested PCR, initially one primer is added and the amplicon of that is used as a primer for the second reaction. In multiplex PCR, where many primers are added together and it can be even done on a real time. What is real time? Real time is the PCR can be analyzed within 1 or 2 hours and it can be monitored onto the computer where you can see the exponential rise of the DNA molecule if it is present. And this is reverse transcriptase PCR, which is done for RNA molecule which we converted into a DNA and amplification done. Applications of PCR in diagnosis of infections, in diagnosis of inherited disorders, sickle cell anemia, beta thalassemia, cystic fibrosis, in cancer detection, and also in medical legal cases. And non-nucleic acid based analytic method, that is chromatography, where we have two phases. One is the mobile phase and the stationary phase, and where we can analyze the molecules which are not DNA but other ionic molecules. This is the gas liquid chromatography and high performance liquid chromatography. Coming to a short introduction about our Central Research Lab, we have a high performance liquid chromatography in our Central Research Laboratory, where we have done many projects with about beta thalassemia. This is a biosafety cabinet where we collect the DNA. This is the real-time PCR where we are working currently on human papilloma virus for detecting cancer. This is a gel documentation after the PCR is run. This is the fluorescent microscopy, and this is the deep freezer where we store all the extracted DNA for further analysis. These are the current projects, molecular projects in our Central Research Laboratory of our PhD students. Many students are working on antibiotic resistance of many bacteria and fungus in cancer patients and in various organisms, and even in hepatitis C virus, MRSA, and on placenta in hypertensive and diabetic women, and type 2 diabetes, dental caries, and FTO gene in with diabetes mellitus, dermatophytes, and disease association of gallstone with CYP7A1 gene, and prevalence of human papillomavirus, the genotyping study, angiotensin converting enzyme study, and histomorphological changes in placenta of undernourished mother. With this brief introduction, I would like to end my talk. Thank you.
Thank you so much, ma'am, for wonderful introduction.
I think, it's sort of contextual learning. They are actually, in context they are talking. हमारे यहाँ पर क्या है? The knowledge acquired, regurgitated in examination, then forget about it. Which is the wrong way of thinking. When I'm sitting in front of me, in the dentistry for that matter, how much importance biochemistry has got? Really, it has got enormous importance. But how much the students give it, the moment they clear it, they just keep it. Hardly ever they apply that knowledge. Likewise, microbiology also, I had this session, madam. After I became a faculty member, somehow I opened all my books. I had not fortunately thrown them. And microbiology, its clinical relevance, physiology, I just thought, oh my God, this was intended and what I had done. So, in this problem-based learning, everything, this is called horizontal vertigation as well as vertical integration. To apply the knowledge which you have already gained. So I won't be going into details of that. In molecular biology, in immunology, we gave the students a case of AIDS. We gave the students certain problems related to molecular biology, and we found, that is our experience, and we published the results in very esteemed journals also, esteemed international journals also, although we said that this is very much, not as per, the traditional definition of problem-based learning, but the question, but still, they appreciate it, they published it also, but still the question is that we have to see everything in our context, in the Indian context. Now the last thing, the criticism which I have been getting is our cultural attributes. Indian cultural attributes, are they compatible with this type of teaching? हमारे यहाँ पर क्या है? Silence is considered a big virtue, a big quality, and being outspoken runs the risk of being immodest. बड़ों के सामने बोलती नहीं है टाइप एटीट्यूड, लड़की बहुत अच्छी मुंह में जबान नहीं है, so that type of attitude. And then there is fear of confrontation with the authorities. I have seen, many times, mobile is helping in one way. You go anywhere, you can stand anywhere taking your mobile and listen to what the other person is talking. And nobody suspects you. And this I do in various institutions, talking to the students. And I've seen that they talk, nazron mein batao, teacher ke samne bolo nahi chahiye. So with this type of attitude, are we prepared for this way of teaching? I leave this question open, but I have traveled in various places, interacted with other people of various other nations also, but I'm talking about the SAARC nations. It is very well implemented in Pakistan, in Bangladesh, Sri Lanka is going great guns, and the entire teaching of our neighbor, Nepal, is based on problem-based teaching. And this model has been found to be very useful. So, if not fully, partially, this can always be implemented, because I'm returning to molecular biology, immunology, so much of growth is there. Not long back ago, there used to be only one chapter of 20 pages in the textbook of biochemistry, Harvard, replication, translation, and that's all. But now what it has become, it is occupying 50% of the syllabus. And I don't know, madam, in immunology also there is a lot of emphasis on microbiology. Immunology actually, it has been inextricably linked with the viral diseases. हमारे टाइम में तो वायरस का बस, वायरस was something, one virus meant for me 20 marks essay type question, which would be repeated every alternate years. So the experts in that field would tell me, इस साल आप virus मत पढ़ो, अगले साल आ जाएगा नहीं करो. But now the things are really changing, and I just based on my personal experiences, based on the existing literature, I have been able to, I think, communicate about the poetry, about the problem-based teaching, etc., particularly with relevance to molecular biology and immunology. Thanks a lot.
Thank you so much, sir.
The success of the talk is reflected by the questions if they come at all.
Thank you so much, sir, for your submission. I request training ma'am to please give token of appreciation to Dr. Dinesh sir. I request Saraswati ma'am to please give token of gratitude to chairperson Dr. James ma'am. I request Saraswati ma'am to please chair the section for starting the second section. I request ma'am to please invite our chief guest Dr. Bushra Singh ma'am to deliver her lecture.
It gives me immense pleasure to introduce one of the great scientists, Dr. Bushra Singh ma'am. She has obtained a PhD from Aligarh Muslim University, Aligarh. After a brief stint as a research associate at the Olivier Institute of Medical Science, New Delhi, and National Institute of Immunology, New Delhi, she pursued her postgraduate training from McGill University, USA. Later, she also served as a research investigator as a junior faculty at the Migrant Center for Translational Pathology. She has been the recipient of a number of prestigious national and international awards. To name a few, Fast Track Young Scientist Award by DST Sir, Ramanujan Fellowship Award for DST, AACR Women in Cancer Research Scholar Award from American Association of Cancer Research, Young Investigator Award from Inspiration Fund for Breast Cancer Research, and Genitech Postgraduate Doctoral Award from the Genitech Foundation, South San Francisco, USA. It's a great pleasure to have you around. Thank you.
For inviting me to deliver a talk here. It's like my pleasure to be here at Rama Medical Institution. And so I thought like I'm going to talk about more on this, like teaching and all, dog tricks, repositioning or drug repurposing. It's about like malaria drug, artificial selection, prostate cancer. And I thought like it's so. Before I go to the main topic, I'm going to just brief you about stress, what exactly I do, and a little bit about prostate cancer in general, what we did in the past. So, my research interest revolves around in addressing biomedical problems which are related to biomedical research, the problem which have clinical significance using some innovative approaches and techniques as Dr. Puri mentioned, and look for the problem which has translational impact. So, today I'm going to talk about only prostate cancer. So basically prostate cancer is a localized disease of prostate gland. Initially when it started, it is responsive to the anti-androgen therapy. But over a period of time, you see that the patient will progress to develop metastasis, and at this stage, it's known as CRPC, castration-resistant prostate cancer, and at that stage, patient no longer respond to anti-androgen therapy. So basically in India, it's like by default, like we give patient anti-androgen treatment that is also known as androgen deprivation therapy. And some patients respond, whereas other patients don't respond. So it's very important to do a molecular subtyping. Divide the patient based on the molecular alteration or whichever molecular alteration. When I say it's like including mutation, genetic rearrangements. So basically they serve as a driver of the cancer. So here you use different techniques. You can do NGS-based cancer genome to explore what are different mutations or what are different genetic rearrangements in those patients. Perform a FISH technique where you have a fluorescent probes and look at the arrangement of that particular genetic arrangement. Then simply do IHC, immunohistochemistry, for the known oncogenes, and that's how you divide the patient. So in the lab, what we do characterization, plus we look for the functional relevance of these molecular alteration or mutations, look at the mechanistic pathways which are involved in driving the oncogenesis, and then also do the drug screening. This is androgen responsive, means when you give anti-androgen therapy, they respond. But over a period of time, the patient they stop responding to anti-androgen therapy. And at the genetic level, you see loss of many tumor suppressor genes, for example, NKX1, P10, Epiblastoma. These are some tumor suppressor genes which show deletion or hypermethylation, for example, hypermethylation. While many oncogenes such as GSP1, heparinase, NSCR, they are upregulated. So this is the nutshell. But again, when you want to see what are the molecular drivers in prostate cancer, there are distinct about. This is the first genetic alteration that was discovered. In this genetic alteration, in this genetic arrangement, the TMSS2, which is the androgen-driven gene, apparently comes closer to our gene, which is silent in prostate in the normal gland. And because of this aberration, where the 5' partner, the first exon of TMSS, apparently fused to the ERK gene, the ERK is overexpressed. Now here, this genetic treatment is present in 50 to 60% of the prostate cancer patient. So now, in this context, when I say ERK is acting as a oncogene, it is overexpressed in 50% of the Caucasian population. Another subtype is SPINK positive status. SPINK is a serine protein in human carbon fiber, which is overexpressed in 10 or 15% of the prostate cancer patient. And these patients, they show a very aggressive disease because they have a shorter biochemical regularity, which means the duration for PSA, which is a prostate-specific antigen, to go up is reported to be shorter than the positive patients. And these two subtypes, they are mutually exclusive, meaning a patient who is positive for TMSS2 fusion is negative for SPINK1 and vice versa. Another subtype which belongs to Indian prostate cancer patient, and here is the IHC. I'm sure most of you know what an IHC is, immunohistochemical staining. And then this treatment was confirmed by performing FISH, which is a fluorescent in situ hybridization. So both probes here are located in the ERK locus, and if a cell is having a no fusion, like no genetic arrangement between the ERK and TMSS2, you see a signal like this, green and red merge. But if there is a genetic arrangement between TMSS2 and ERK, you see two signals going away. So break apart which was performed to detect this fusion. And only another subclass is EDP1, only one patient was found positive for EDP1. We also look for the overexpression of SPINK1, which is a serine protease inhibitor peptide. As you remember, it's second highest alteration in Indian cohort as well, similar to patient. And this staining for SPINK1 is very specific. This is a sample from prostatectomy, robotic prostatectomy, around 4+4 patient, which is very distinct staining for the prostate cancer foci. And here you see this is a little biopsy, again very specific for SPINK positive subset. Then we will also look for the status of the tumor suppressor gene, P10, which is very commonly mutated in prostate cancer patient. We found about 18% of the patients they have they have the normal copy of the P10, and maybe the more than 2 copies are there. There is a heterozygous homozygous deletion in 10 and 11% of these patients. What was interesting to see was most of the patients who have genetic arrangement between us also have a deletion of P10 at the tumor suppressor gene, and those patients show much more higher disease score, which is an indicator of how aggressive the disease is. And here the way FISH was performed, you take two probes. This probe is in the central region on chromosome 10, because P10 is located on chromosome 10, and then P10 this gene is located there in another probe here. So, for example, if you see a normal gland, like a normal cell, copies green, 2 copies red. Obviously, it was two set of chromosomes. But if you see 1 copy lost, you see 2 green because 2 pair of chromosomes and only one set of red. So 1 copy is lost. Likewise, if you see only green, then both copies are lost. So we also look for the RAF genetic variant, which is, and we found that it's much higher in Indian prostate cancer patients than Caucasian. So the number in Caucasian is only 2%, whereas the Indian population is 4.5 and around 5% for RAF and BRAF penetration. And most of these patients, they show much more aggressive disease, that is 7 to 9 weeks is more. So we published this report. It was the multi-institutional research. But here you can see this is the overall statistic that we found in context of Indian report. The good news is that most of the patients who harbored this RAF genetic engagement could respond to FDA-approved RAF inhibitor which are already there in the clinic, and the percentage in the Indian population is 6, whereas the Caucasian is only 2. So having said that, now I switch my gears to CRPC. So besides having all these molecular subtypes like Spain and RAF, and most of these patients, they are finally progressing into a much aggressive disease that is like castration-resistant prostate cancer. At this stage, they get like first line or second line pharmacotherapy. CYP2, not many patients they receive it. This is a immunotherapy, but despite of all that, they finally progress to castration-resistant disease. And here they are given this abiraterone, which is a CYP inhibitor, or enzalutamide, another anti-androgen. Most of these patients, they won't respond to these anti-androgens, and they develop resistant CRPC, a disease which is much more resistant to these anti-androgens. And after that, eventually there's no cure. So at the molecular level, you want to see what is happening in this disease because, as I mentioned, the phenotype is much more aggressive. So at this stage, when they are localized and they are having they are responding to castration therapy, you see AR is high, that is androgen receptor. There is a rise in PSA, which is the indicator of the disease, and there is some activating mutation in the AR receptor. Activity mutation, then the signaling will be in the ligand-free, ligand-independent manner, and then there are certain splice variants which are much more prominent in these patients. And beside prostate gland, there is a synthesis of DHT, this is high dihydroxytestosterone coming from the adrenal gland as well. But once they come to this stage, they bypass the androgen signaling. They are again androgen receptor positive status, but glucocorticoid receptor will be upregulated. Finally, at the complete AR independence, you see most of the patients, they show low AR, low PSA, but the histological features are different than these two subtypes. Here they are more NPC, that is neuroendocrine prostate cancer, also known as small cell carcinoma. And these subtypes are relatively new. A few years back, these have been in use. And at this stage, you see a lot of other genetic alterations. So what we want to know here, at this stage, these patients, they acquire resistance to ADT, that is androgen deprivation therapy. So one of the features of this ADT is NF-κB signaling was highly upregulated, and NF-κB signaling is known to associate with high incidence score. They are much more having an incidence of biochemical recurrence, shorter biochemical recurrence, and shorter disease-free survival. So there's two types of NF-κB signaling, chemical pathway and non-chemical pathway, but both these pathways merge and there is a crosstalk between NF-κB, that is P50, P65 and androgen receptor. So in a CRPC patient, NF-κB activation occurs as the AR loses the ligand binding domain, or there is a mutation in the ligand binding domain, and then that's how the NF-κB signaling takes over, and this secondarily activate the AR during transition of CRPC. So the idea here was if we block the NF-κB signaling pathway, and what we thought, like maybe it would use one of the established drugs in the market. So instead of going to the semi-synthetic drugs, like there are many NF-κB inhibitors, but there are many reports where people have shown these patients, they show off-target effect and there is toxicity issues related to those NF-κB inhibitors. So we thought we screen the molecular drugs which is well known repurposing is also known as the reprofiling of a drug or repositioning. So what you do here, you take the old drug which are FDA approved or WHO approved and see if that old drug could be used to treat a new disease or other disease or other molecular pathway. So if you look at the graph here in the drug discovery, from discovery to the FDA approval, it takes 12 to 15 years for a new drug. But if you go to drug repurposing, it's only like 4 to 5 years, and the money you spend is much lower than the money you spend on a new drug. So there are various ways that people use for drug repurposing. You can do a omics approach like proteomics, genomics, or metabolomics approach. You can go through published articles and see what are the targets. You look at the mutation and genetics behind the disease, phenotype and the structure. You can do computational docking. You look at the structure of protein and see which drug will fit in the docking in the active domain, and then that's how you move to the drug repurposing. So if you remember, last year Nobel Prize was given to this lady, Yu in physiology for the discovery of artemisinin. Artemisinin, as you know, is food is for the third world country which have millions of malaria cases. So the artemisinin drug is derived from the skin cow, Artemisia annua. The scientific name is this, and this is a semi-synthetic drug and well known for its use in malaria. Even for the resistant malaria patient who have resistant malaria, it's used as a combination therapy. So what we thought, like maybe we would see the effect of the malaria drug on the prostate cancer cells, and the molecular mechanism of the artemisinin is well known. It was already established. It has a very unique structure. If you see here, and there's an endoparasite bridge here, and because of the presence of the ferrocyanine, this will attack the endoparasite bridge and produces the ROS, reactive oxygen species, and that's how it will cause the lyse of the malaria parasite. And same thing could be recapitulated for the cancer cell that was we thought, because most of the cancer cell also have high ferrocyanine. There are like receptor transferins which are highly upregulated in cancer cells, which will facilitate the uptake of the ferrocyanine. And if you treat the cells with this artemisinin, these cells will undergo apoptosis. So that was the idea behind this study. So what we did, we used 4 different form of like semi-synthetic form of this malaria drug: artesunate, artemether, artemether, and dihydroartemisinin. And PIP is biocombat anti-androgen, which is commonly used in the clinic for prostate cancer patient. So you can see the cell line that we use here, PC3 and gamma-2R1 cells. PC3 and 2R1, they are much more aggressive cell lines. They are like much more like a castration-resistant kind of cell line, and they are least responsive to anti-androgen therapy, whereas this LN-CAP is a responsive cell line. It's AR positive, and you see here these are the IC50 value for artesunate. We found other derivative of the artemisinin. They are not very effective in our cell lines, so we stick to artesunate. Then we performed this isohologram to see what is the best combination of these 2 drugs. We found that PC3 and 2R1 cells, they show a very strong synergistic interaction, because CI is a combinatorial therapy index. If it is less than 1 is much closer to 0, then it's much more strong interaction, and if it is closer to 1, then it's the least synergistic interaction. And obviously these 2 lines show much more stronger synergistic interaction with artesunate and biglutamide is active. So in most of my experiments, we have used the same concentration that I mentioned there. So here you can see we perform a cell proliferation assay to see what is the effect of artesunate, whether artesunate alone could cause cell death, or in combination of biglutamide it can cause cell death. So this is a traditional WST assay, and you can see in the combination therapy, artesunate and PIP, they are much more effective in causing the cell death or in arresting the cell proliferation. Then we asked, like, because cancer, it has a phenotype of invading the ECM, that is extracellular matrix. So usually cancer cells, they secrete urocanase receptor or CLAW, MMPs, which will degrade ECM, and then you see the cells invade in the surrounding area. So in this assay, we treated cells with again combination of PIP and artesunate alone, and again the combination therapy showed much more effective inhibition in PC3 and 2R1 cells as well as LN-CAP. combination with bicarbonate NF-kappa basically because that's how we started. So what we did here, we treated two variables with NF-kappa which will activate the NF-kappa basically, and then we treated the cells with artesunate, bicarbonate and combination together. And we found that at the combination therapy of combination treatment of artesunate and PIC causes decrease in the full length as well as variant E5 AR in cellular share, and then here you see the decrease in phospho 65 milligram. Then we also want to see what is happening at NF-kappa basically. So we did a Western blot for phospho P65, there is significant decrease when we treated these cells with artesunate alone and in combination again there was certain decrease was robust. To recapitulate the same experiment by using a different type of assay, we did a luciferase promoter assay for NF-kappa B promoter. And usually, like here, you are seeing a readout of the NF-kappa B signal, but here it will prove whether the transcription factor which bind the NF-kappa B promoter region, they are indeed binding or not. So, in this luciferase promoter assay, you see this E11 promoter was used, and here there are 5 NF-kappa B binding sites. So if the NF-kappa B signal is active, you see increase in the luciferase activity, while when you use artesunate or artesunate plus bicarbonate, which will inhibit the NF-kappa B signal, you see reduction in the luciferase activity. So as anticipated, we found significant decrease in the NF-kappa B signaling in this reporter assay when a combination of artesunate and bicarbonate was used. Now, the whole idea was to see whether artesunate and bicarbonate will inhibit the androgen receptor target genes, because androgen receptor acts as a transcription factor and it has many genes which are under the control of this receptor, and one such example is PSA, prostate specific antigen. So, what we did, we treated the cells with ethanol because this synthetic androgen was dissolved in ethanol. So, this serves as a control, and this synthetic androgen was used to stimulate cells, prostate cells, and then we use artesunate or PIC either alone or in combination. And as expected, when we use artesunate with PIC, there is a significant reduction in the expression of PSA in both cell and 2R1 and LN-cap. So LN-cap is androgen responsive, so the fold increase you see in the PSA is much more profound than 2R1 cell, which is less responsive to androgen treatment. Then again we perform a reporter assay. So again I want to give recapitulate what exactly we did here for the reporter assay. Here the assay, in this assay what we did, we include the PSA promoter region in luciferase vector, and again when we treated the cells with synthetic androgen which is acting as a ligand, you see the ligand will go and bind to the androgen receptor. Now this androgen receptor in the activated form will go inside the nucleus and bind the promoter region of the PSA, because PSA is the air target gene, and then you see increase in the luciferase activity. So this is another new way of showing that indeed when we use artesunate and bicarbonate in combination or alone, you can see significant decrease in the promoter activity of a androgen gene, that is PSA. Now next we want to see what is the mechanism why like this there is a decrease in the androgen receptor expression. So as I mentioned earlier, when we treat soluble cell and LN-cap with bicarbonate or add artesunate, with a significant reduction in the full length AR receptor, AR or variant receptor pull-down experiment, where we treated the cells with artesunate and PIC and also put MG132 which is a proteasome inhibitor, so it will stop the degradation of the AR if it is inhibited. So for pull-down assay what we did, we used two antibodies, IgG mark, when we treated these cells with artesunate and PIC as compared to ethanol or DMSO control. Likewise, when we did a Western blot for AR receptor, we see there is a significant increase in the AR. It's not antidegrading because we have used MG132, but this was sufficient enough to indicate that artesunate and PIC indeed increasing the implementation mark of the AR, and AR is under 26S proteasomal degradation, and that's how AR expression was going down. Then we perform immunofluorescence assay so that we can see at the cellular level what is happening. And again, here we use PST, which is a dihydroxy testosterone, active form of the testosterone. We stimulate these cells with this and then put artesunate alone, bicarbonate or artesunate and bicarbonate combination. So you can see here, when we treat with PST, you see a lot of green staining in the nucleus, and because AR was configured with a green fluorophore, so AR is moved into the nucleus to act as a transcription factor. While when we put artesunate, there is a decrease in the AR expression, while because there is a significant decrease, and the combinatorial treatment was much more effective than individual one. Next was to see, because one of the properties of artesunate is to induce, to generate ROS, as I mentioned earlier. So here we perform PCMTA assay, and this assay is very simple because there are a lot of cellular esterases inside a cell. So when we use this compound with a non-fluorescent H2BCl, and these cellular esterases, they act on this and convert into non-fluorescent form. And if there is a ROS in the system, or simply if you treat cells with H2O, it will generate ROS, and this ferrous ion will convert it into a DCM, which is a fluorescent substrate. So this assay was performed in these three cell lines, and as expected, when we use a combination of artesunate and PIC, we observe there is significant increase in the fluorescence which is coming from this DCM, indicating that when we treat cells with PIC and artesunate, the ROS division is much higher than the individual treatment. Again, to see what is the effect of PIC and artesunate on cell death or apoptosis, we performed a Caspase-3 assay, because Caspase-3 is one of the major key exhibition of Caspase cell death, Caspase-dependent cell death. So this is a result from Alexa assay. You can see when we perform this assay in the presence of artesunate and PIC, there is a significant increase in the Caspase-3 activity in all the three cell lines. And then we perform Bayesian plot for some of the key players in the apoptosis pathway. For example, clean PARP is one of the readout for cell death, and doxycycline was used as a positive control. You can see here in PC3 cells, it's not very well, but here in human cells and LNCAP, a significant increase in the clean PARP was observed, and as cell survival was gone down when using combination in all the three cell lines. There is no significant effect on the expression of BCL2, BCL-XL was observed. So in nutshell, it indicates that artesunate plus bicarbonate triggers apoptosis cell death in prostate cancer cells by inducing oxidative stress and activating Caspase-3 activity as well as cleavage of the PARP. Then finally, we performed xenograft, prostate cancer xenograft assay. Here we use PC3 cells and implanted these PC3 cells in nodal-state mice, which are immunodeficient mice. So the mice were randomized and divided into 4 different groups. Control, who received like just BTS or saline, then artesunate group which received like artesunate, bicarbonate received group, and a group which received the combinatorial treatment. And we found every alternate day, the tumor burden was measured by using the digital Border calicos and was calibrated as spotted here. So you can see the mice treated with either control or artesunate alone or bicarbonate did not respond at all, while the mice who received a combinatorial therapy with artesunate and bicarbonate show regression in the tumor burden. Then we also want to see like what is happening in the tumors. So these tumors were taken out, fixed, and the slides were prepared to do a Ki-67 staining, which is an indicator of cell proliferation, and found the mice who received artesunate and PIC treatment show much lower percentage of the Ki-67 positive cell as compared to mice who received just artesunate or bicarbonate. Finally, we were interested to see what is happening, whether this combinatorial drug is inhibiting distant metastasis or not, because metastasis is one of the problems that prevails among the CRPC patients. So what we did here, at the end of the experiment, remember like the experiment was terminated at 27th day. So what we did, we dissected the mice and then took out the tibia, lungs, and then the DNA was prepared. From the DNA, the bone marrow was flushed out. The DNA was isolated from this flushed out bone marrow cells and lungs. And the genomic DNA was used to perform quantitative PCR for ANU sequences. Why ANU sequences? Because in human, you have this ANU repeat But in mice, you have B1 and B2 repeats. So what is happening? We implanted the human cells here, BC3 cells, if you remember. And now the cells metastasize to these organs, that will be detected by performing this allo-PCR. And they will this coordinated PCR. And this is the data we converted our CV values in the number of cells by plotting the graph in another calculation. So you will see here, when we give mice artificial and biochemical treatment, the number of the cells metastasized to a